A very good day, everyone. My name is Lushini and my matrix number is A208966. Today, I will be presenting a research article entitled Tilapia as a model fish for biomonitoring of metal pollution in dams associated with mining watersheds. This presentation is prepared for the course STAE 2443, Scientific Communication in Environmental Science. So, throughout this presentation, I will explain the introduction, its objective, methodology, key findings, discussion, conclusion, and finally the acknowledgement. Let's begin with the introduction. Mining activities are one of the major sources of heavy metal pollution in aquatic ecosystem. During mining operations, heavy metals such as cadmium, copper, lead, and zinc can be released into the nearby rivers and reservoirs and also the dams. These metals remain in the environment for a long time and gradually accumulate in aquatic organisms. Among aquatic organisms, fish are particularly important because they are a part of the human food chain. When fish are exposed to contaminated water, heavy metals accumulate in different organs and tissues through a process called bioaccumulation. In this study, the researchers selected tilapia because it is widely distributed, commonly consumed, and highly tolerant to environmental stress. More importantly, tilapia reflects the environmental conditions of the water it lives in, making it an excellent biomonitor for detecting heavy metal pollution. The study focuses on 11 dams located in northwestern Mexico, where the surrounding watersheds are influenced by mining activities. The study had three main objectives, where the first one is to determine the concentration of four heavy metals, namely cadmium, copper, lead, and zinc, in different tissues of blue tilapia collected from 11 mining-associated dams. The second objective was to compare the level of metal accumulation among these dams in order to identify which locations were more contaminated. Finally, the third objective was to evaluate whether tilapia can serve as an effective biomonitor for monitoring metal pollution in freshwater ecosystem. Now, moving on to the methodology. The research was conducted in 11 dams located in Sinaloa, northwestern Mexico, within the southeastern Gulf of California region. These dams experience different levels of mining influence, making them suitable location for comparison. A total of 320 blue tilapia, scientifically known as Oreochromis aureus, were collected during two different sampling periods. The rainy season sampling periods took place from August to October 2015, while the dry season sampling was conducted between February and March 2016. After collection, the researchers dissected four different tissues from each fish. These were the muscles, liver, gills, and the guts. The samples were then analyzed for four heavy metals, which is cadmium, copper, lead, and zinc, using atomic absorption spectrophotometry or AAS, which is a highly accurate laboratory technique for measuring metal concentration. To compare the data statistically, the researchers used the Kruskal-Wallis test, Mann-Whitney U-test, and the Spearman correlation analysis. Finally, they evaluated the health risk associated with consuming tilapia mussel using three internationally accepted risk indicators, which is the target hazard quotient, hazard index, and the cancer risk assessment. Now, let's look at the results beginning with the muscle tissues. The researchers found that metal concentration in muscles varied among the 11 dams. Among four metals, the zinc showed the highest concentration, while the cadmium showed the lowest concentration. These dams with a relatively higher metal concentration were dams 1, 2, 5, and 10, suggesting greater environmental contamination in these locations. Although mussels is the edible part of the fish, it accumulated much lower concentration of heavy metals compared to liver. This is encouraging because muscle tissue is what humans usually consume. Most metal concentrations remain below the legal safety limit. However, lead still poses a potential health concern, especially for people who frequently consume tilapia from contaminated areas. These findings suggest that contamination differs between location and is likely associated with mining activities surrounding the dams. Next is the liver, which showed the most significant finding. Among all four tissues, the liver accumulated the highest concentration of heavy metals, particularly copper and zinc. This is expected because the liver is responsible for detoxification, metabolism, and storage of harmful substances entering the fish's body. The highest liver metal concentration were recorded in dams 1 and 2, indicating that fish from this location experience greater long-term exposure to heavy metal pollution. Another important observation was the highest accumulation of zinc in several dams. This suggests continuous exposure to bioavailable zinc, likely originating from mining activities. Because of its highly accumulation capacity, the liver proved to be the most informative tissue for environmental biomonitoring. The third tissue analyzed was the gills. Unlike the liver, the gills are directly exposed to the surrounding water throughout the fish's life. The study found that zinc was again the dominant metal in this tissue. Metal concentration varied among the different dams, indicating differences in water quality and levels of contamination. Some mining affected dams showed noticeably higher metal concentration in the gills. This indicates that the gills are particularly useful for monitoring recent or short-term exposure to contaminated water because they are the first organ to come into contact with pollutants. The fourth tissue examined was the guts. The guts represent exposure through feeding because tilapia consumes sediments, algae, and other food sources found within the aquatic environment. The study found relatively high concentration of zinc and copper in the gut tissue. Again, metal concentration differed among the dams, suggesting difference in sediment contamination and food availability. Compared with muscle tissue, the guts accumulated higher concentration of metals, demonstrating that ingestion is another important pathway of heavy metal uptake. Moving on to the discussion, one of the most important findings of this study is that tissue reflects a different exposure pathway. The liver reflects long-term storage and detoxification. The gills indicate direct exposure from contaminated water. The guts reflect contamination through food and sediments. Meanwhile, the muscles represent the edible tissue and it is therefore important for assessing risk to human health. The study also showed that dams 1 and 2 consistently recorded the highest contamination levels, which were associated with intensive mining activities and possible geothermal influences. Although mussel concentration generally remained within food safety guidelines, lead was still identified as the greatest health concern, especially for children and people who regularly consume fish from contaminated areas. Overall, the findings demonstrate that examining multiple fish tissue provides a much more complete picture of environmental pollution than analyzing muscle tissue alone. To conclude, this study successfully demonstrated that tilapia is an effective biomonitor for monitoring heavy metal pollution in freshwater ecosystem. Metal accumulation differed depending on the tissue, the dam, and the sampling season. Among the 11 dams, dams 1, 2, 5, and 9 recorded the highest contamination, while dam 11 showed the lowest metal concentration. The liver was identified as the most informative tissue because it accumulates the highest concentration of cadmium, copper, lead, and zinc. Although the edible mussel generally contain metal concentration below legal safety limits, lead remained a concern, particularly for children and frequent consumers of tilapia. Therefore, combining tissue analysis with human health risk assessment provides valuable information for environmental monitoring, pollution management, and food safety. The information presented today is based primarily on a research article by Paes Osuna and colleagues together with supporting references on tilapia biology and environmental tolerance. Finally, I would like to acknowledge the authors of the original research article, Paes Osuna and colleagues whose work formed the basis of this presentation. I would also like to recognize the collaborating fishermen from the 11 dams in Sinaloa for assisting with fish sampling and data collection. In addition, appreciation goes to H. Bonjour Levia for analytical support, as well as the funding agency including GPA, PAPIT, and Universidad Nacional Autónoma de México, CONAIEC, and the Federación de Cooperativas Pecuarias de Sinaloa for supporting this important research. Thank you very much for your attention. That concludes my presentation and I appreciate your time.